Okay, good morning everyone and welcome to the 2015 edition of the Royal Tyrrell Museum Speaker Series. Today the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Julie Meachen. Julie is an assistant professor in the Department of Anatomy at Des Moines University in Iowa. Julie is originally from Tampa, Florida. She obtained her bachelor's degree in biology at the University of Florida in Gainesville. She decided to stay at the University of Florida to pursue her master's degree in paleontology. For her thesis, Julie described a new species of llama from the Paleoplastocene and did a comparative analysis of llama functional morphology. After completing her master's degree, Julie moved to California to pursue a PhD in the Department of Ecology and Evolutionary Biology at UCLA. For her dissertation, she examined the morphological differences between large and small felids and how these relate to prey-killing strategies. After completing her PhD, Julie held a postdoctoral fellowship at the National Evolutionary Synthesis Center in Durham, North Carolina, and taught for 1 year at Marshall University in Huntington, West Virginia, before becoming a professor at Des Moines University. Julie's research interests focus on the functional morphology of large mammalian carnivores, particularly from the Rancho La Brea tar pits, and what it can tell us about major environmental changes. She is currently leading excavations in Natural Trap Cave, Wyoming, studying the remains of late Pleistocene mammals found at the site in order to determine how morphology correlates with ancient DNA and climate change. Today, Julie will discuss recent work she conducted with her students on coyote ecological changes since the last ice age and talk about her research at Natural Trap Cave. So without further delay, I present you Dr. Julie Meachen.
Thanks, Francois. That was a great introduction. Thank you. All right. So I'm going to talk to you a little bit about what I study. Francois did a really nice introduction. And then I'll tell you about what I'm going to talk about today and how it all relates to one another. So my research program, when I study fossils, I really like to ask the question, how can we use functional morphology and fossils to examine patterns of environmental change through time? And I like to do this with my favorite group of animals, the mammalian carnivores. So this talk will mostly be about them. There will be a few other critters when we talk about Natural Trap Cave. And why mammalian carnivores, you may ask? What is special about them? Well, besides being just altogether cool animals, they're actually really important for the environment. They do top-down control of all the other animals in the environment, and they keep the environment in the conditions that it should be in. And you may have heard some things in the news within the past 10 years or so about wolves in Yellowstone. And once they were reintroduced, they actually changed the environment from what it was when they were missing. And also sea otters and the Pacific kelp forests on the west coast of North America. And how otters actually keep those kelp forests healthy by keeping the sea urchin populations in check. So here's just an example of some things that carnivores do for the environment. On the right side, there's some pictures of environments that have carnivores in them. And on the left side, there's pictures of environments that don't have carnivores in them, and you can actually see how different they are. So they don't just control, they sort of control everything, including vegetation from a top-down perspective. And they live in just about every kind of habitat in the world, and they're native to almost every continent except for Australia and Antarctica. So they're everywhere. So with that, and telling you why I study carnivores, I'll tell you a little bit about what I am going to talk to you about today. So the first thing I'm going to touch on, or go into a little bit of detail on, is how the Ice Age extinction event affected a species that was able to live through it. And this is coyotes. And I'll discuss some work that I did a couple years ago and a paper that I just put out this January along with some of my students that build upon that former work. And then I'll talk to you about the site that I'm currently excavating, Natural Trap Cave, and what we can learn about it. And then finally, I'll go into a very small amount of detail about some of the wolves from Natural Trap Cave and the Yukon. So there'll be a little bit of Canadian biology in here too. And some modern localities and what we can learn about those groups of organisms. So let's go ahead and start with the Ice Age extinction event. So a question that I really like to ask is, how did the megafaunal extinction affect species that lived through it? So we all know that lots of different big mammal species went extinct at the end of the last ice age, but most people don't actually think about how that extinction event affected the species that are still around today. And the species that I wanted to focus on for this question is the coyote. And the reason I wanted to do that is because we have really great numbers of coyotes in the Pleistocene, and we also have really good populations of coyotes today. So this is a species that has great abundance and variety data before and after the extinction event. So this is a great species to ask this question. So this is a modern coyote. And modern coyotes, when they live in habitats with gray wolves, generally have some competition. And this is important for the talk because it basically shows how this competition affects these animals. And so when we put them together in places like Yellowstone, there's often a lot of actual fighting that goes on. There's some poor coyote fatalities. Oftentimes wolves will just kill coyotes for sport. They don't like them. They're a competitor. They're slightly smaller, so they get picked on. And in the Pleistocene in North America, we had this species that lived here called the dire wolf, which was about a third again as large as the living gray wolf. So if we're looking at the Pleistocene and we're wondering how coyotes have changed since then, we got to think about these guys being the coyotes' competitors. So if gray wolves and coyotes don't get along, what is going to be the situation with the poor coyotes in the Pleistocene? Are they going to be at a disadvantage? And to answer this question, I go to one of my favorite fossil localities. This is Rancho La Brea. It's actually in the middle of Los Angeles. So this is what the outside looks like right here. Here's this big pool out front that has these nice methane bubbles coming up, which is kind of what it would look like in the Pleistocene for these animals that got trapped. And then we have this life-size diorama of this mammoth family with, looks like mama mammoth or papa mammoth dying and baby and the other looking on here. So this is, you know, a little artistic license, but maybe this is actually what happened. And what would happen is that this herbivore would get stuck in this watery pit coming to take a drink, and then it would attract the attention of multiple carnivores, which would all come running in order to get an easy meal. But unfortunately for a lot of those carnivores, they themselves would become trapped. So the ratio we get at Rancho La Brea is about 10 carnivores for every one herbivore. And this is, I like this picture because this is basically like right across the street from the La Brea tar pits. So you can see that it's actually in the city. It's not like out here where you actually have to drive a little way to get to the nice fossil beds. It is right in the middle of the city. And one of the most abundant fossils at the tar pits is this guy, a subspecies of coyotes, Canis latrans or cut-eye. And it's actually the third most common fossil at the tar pits, in behind dire wolves, which are number 1, and Smilodon fatalis, the saber-toothed cat, which is number 2. And that's important for later. So how were the Pleistocene coyotes different than their living counterparts? And what effect did the Ice Age extinctions have on a species that's still around today? So to answer this question, in a previous study in 2012, I took postcranial measurements of all the coyotes I could get my hands on at Rancho La Brea. And I tried to see all the coyotes I could get, and then I also took measurements from modern coyotes to compare them to. And this is the recent work I've done with a student. I also took photos of coyote jaws in the lateral view, and I placed landmarks on these photos. I wanted to get an idea of how the shape of these jaws has changed over time and what functional signal we could get out of these changes, if there were any. And so I did some analyses on this data, on these data. So I did some postcranial one-way ANOVAs on all the limb bones. And for the mandibular analyses, I did 2D geometric morphometrics with a Procrustes analysis, and I really love this. Basically, does anybody know who Procrustes was? Anybody know their Greek mythology? Yeah, so Procrustes was an innkeeper who used to stretch his victims if they were too short for the bed and chop his victims off if they were too long for the bed. And so they named this technique after him because basically what you do is you take a morphological character, you take a lot of them, and you either stretch or shrink them to follow a consensus form so that you can see how the shape differences are changing without taking size into account. So they named this analysis after Procrustes, and I really like that. So I thought that was interesting. So this will give us a good idea of what shape differences there are between those mandibles without necessarily worrying about size, although you can get size from this, this test as well. And so for the results here, this is the results from the postcranial data. This was in my 2012 paper. These dark boxes right here are all of the Pleistocene coyotes that I measured. So I measured them from a couple other sites besides Rancho La Brea. But what you can definitely see here is that these Pleistocene coyotes are much bigger than all of these other modern coyotes and even some of the Holocene things that were sort of pre-European settlers. So there is something going on between the Pleistocene and the Holocene. These coyotes are changing. And femur circumference is a really great proxy for the mass of these coyotes. Since it's a weight-bearing bone, you can get a good idea of how much their size and their mass has changed. And when I look at the results from the jaws, what I see here is that we get 3 different groups. Over here are the Pleistocene coyotes. And this is sort of the jaw that is indicative of that morphology. Over here we have the modern groups, and here's the jaw that's indicative of that. And if you might not be able to see the major changes in them, but I'll tell you exactly what's going on in just a minute. And then down here in this corner, we actually have this group called Pit 10, which is a Holocene population at Rancho La Brea that is a post-Pleistocene extinction event, but pre-European settlement. So Pit 10 has a date of about 10,000 years old. So we actually see that there are changes going on right after the extinction event. And what we see, the major differences, and I'll reiterate these in a second, is that in the Pleistocene we have these more robust jaws. They're actually biggest in the Pleistocene too. They're sort of uniformly thick all along. They're a little bit thinner in the front, but they're fairly thick up there. By the time we get to Pit 10, which is that Holocene pit, what we see is that the jaws are shrinking a lot. The size decreases quite a bit, but all of those other morphological factors stay very similar. And the way we can really see this is this huge carnassial here compared to the carnassial up here. What you notice, it looks really big here, but that's because these carnassials are about the same size, but this jaw has shrunk since the Pleistocene. And then finally, in the Holocene, what we see is we see the jaws actually getting longer, but they're very thin. So we have these really thin, long jaws, and I'll tell you what that means in just a second, more functionally. And so just to give you an idea of a comparison of a Pleistocene coyote jaw with a modern coyote jaw, the red arrows indicate the shearing surfaces, so parts on the jaw that were made to eat meat. The green are the grinding surfaces, so the parts that will be either eating fruits or maybe grinding up small animals. And then the blue arrows indicate the anterior jaw depth. And so if we compare these two, what we see is that we have this huge discrepancy between the grinding to shearing ratio from the Pleistocene to the modern. And then the jaw depths are really different also from the Pleistocene to the modern. And so what we see here is a transition from the Pleistocene, where coyotes are eating a lot of meat. They're not eating a lot of vegetation, it doesn't look like, from the size of their grinding arcade, to the modern, where the coyotes are eating much more omnivorous diet and much smaller prey, so like rabbits, rodents. And I'll talk about this anterior jaw depth in just a second. So why were Pleistocene coyotes different? Well, was it climate? Because climate is a big thing that's changing at the end of the Pleistocene extinction event. So to answer this question, I took coyotes from many different populations in North America. All of these little black dots here indicate modern coyotes. Here is the mean annual minimum temperature. So this is basically testing to see if Bergmann's rule holds for coyotes. And Bergmann's rule states that as you move up in latitude or you get into a colder environment, the larger the animal gets. So is this a climate effect? Because we think it was a little colder in the Pleistocene than it was today. Well, actually, when I did a regression for that, there was no significant regression. So basically that means that coyotes don't follow Bergmann's rule. They don't get larger as it gets colder. They don't get larger as you go up in latitude. And those red squares there, those red triangles, excuse me, those are those Pleistocene coyotes. And you can actually see that even though it was cooler in Southern California in the Pleistocene, it definitely did not get the seasonality that you get in a place like Yellowstone. So those coyotes are not bigger just because of climate. They're bigger because of something else going on. So what else is changing at that Pleistocene-Holocene boundary? Well, extinction. That's a huge thing that's happening. Faunal turnover. So in the Pleistocene, coyotes had many different competitors. There were a lot of big carnivores in the Pleistocene. Also, one of the other things that they could have probably had more of is prey options. And I'm not suggesting that coyotes ate buffalo or llamas or horses or camels or any of these full-grown things, but babies of these mega herbivores could have definitely been easy coyote prey. So could this affect their morphology and ecology? Well, looking into some modern coyote biology, what I found was that modern coyotes can really change their behavior in response to competition. A couple of studies found that when wolves were reintroduced to Yellowstone, coyotes actually changed their social structure in response to this. So coyotes are a fairly behaviorally labile species. And they also found that coyotes that were in packs today will also kill larger prey together, like deer. So I'll give you one more piece of this coyote puzzle. And that is that dire wolves and coyotes are both North American natives. They're both species that evolved in North America together over several million years. They've coexisted in America together, and so they've pretty much gotten used to each other. They probably competed, but they sort of made their own niches. And they probably had worked things out more or less. But we get gray wolves in the picture, and gray wolves actually didn't evolve in North America. They evolved in Eurasia. They came over in small numbers starting in the late Pliocene, early Pleistocene, but really there's not a lot of gray wolf fossils in North America until after the Pleistocene extinction event. That's when we get really large numbers of gray wolves coming over into North America. So dire wolves went extinct, and now coyotes have this new competitor in big numbers, gray wolves. Well, coyotes are really big because they've kind of found their niche with the dire wolves. But we know that when coyotes and gray wolves come into contact with each other, there's lots of conflict. And so what it looks like was going on is that coyotes were big in the Pleistocene, and after gray wolves moved in from Eurasia, they're still bigger than those coyotes. They're still bigger than the biggest coyotes. So it may have been a combination of fewer prey items and also those larger gray wolves basically picking off the largest coyotes, and so making their body size, their median body size, lower in general. So how did being larger affect the coyote ecology? Were they hunting in packs? Well, coyotes are the third most common fossil at the tar pits. And there was a study that came out a couple of years ago that suggested that Smilodon fatalis, a saber-toothed cat, was social based on their numbers at the tar pits. I'm happy to talk more about that later, but it'll probably just get in the way right now. But if you have questions on that, I'll be happy to answer them. So if larger numbers of Smilodon suggest that they were social, it stands to reason that larger numbers of coyotes also suggest that they were social in the Pleistocene at the tar pits too. Were they chasing more prey? Well, we know that they were probably eating more meat, but a lot of people have said to me, well, weren't they scavenging more? And when I did the first study in 2012, I couldn't answer the question whether they were just scavenging more or whether they were actually hunting their own prey. But the jaw study that I did, remember I told you they have those larger anterior jaws? That's important. Because, here we go, anteriorly robust jaws are actually indicative of higher forces on the jaw of large canids that hunt big prey, like wolves. Because basically their strategy is when they're chasing prey, they basically run into the rump of the prey and they nip them. And they do this, well, they chase them to wear them out. And so what happens is that anterior part of the jaw gets built up more, and it looks like this is what was going on in those Pleistocene coyotes. So the anterior part of their jaw is thicker, which suggests they were actually hunting larger prey. You don't need a thicker anterior jaw to hunt small prey. You need a long jaw, which is what they have now, because that makes your jaw close faster, speedier jaw, but that thicker jaw, that's indicative of eating bigger things. And the coyotes in the Pleistocene actually also have higher frequencies of tooth breakage and wear, which suggests that they're definitely eating more bone or that they are competing more with each other for food resources. So how did the megafaunal extinction event affect this modern species, the coyotes? They became a lot smaller. They actually switched their niches from eating slightly larger prey, more meat in their diet, to smaller prey, more omnivorous diet. And they went from probably hunting in packs together to becoming slightly more solitary. So I guess the conclusions of this study is that extinction events don't occur in a vacuum. When we think about extinction events, we only think about the species that actually went extinct. We don't think about the ones that are surviving today. But extinction events affect all living organisms in an interconnected ecosystem. So everything is really interdependent upon each other, and an environment or an ecosystem that's been around for millions of years is going to be affected anytime one of the members or several of the members goes extinct. So they survived the Pleistocene extinction event, but not without significant changes. And coyotes today are continuing to evolve and learn new tricks. There are huge sample sizes, huge populations of coyotes that live in cities now, and there's actually a whole project going on at the Ohio State University that looks at urban coyotes in Chicago. And coyotes are learning new tricks like shopping at Quiznos and riding on the subway. So maybe in 100 years, coyotes will look very different. The next step in this project actually is to start looking at some of the ancient DNA of coyotes from the Pleistocene and comparing it with modern DNA from coyote populations to see if we have had any loss of variation, genetic variation in coyotes between the Pleistocene and the recent, to see how that extinction event actually has changed their genetic makeup. So did they undergo a bottleneck, a genetic bottleneck between the Pleistocene and today? It's one of the questions I'm hoping to answer. And so following from this kind of study, I'll talk to you about Natural Trap Cave, which is my current fossil site. And the reason why these two studies segue nice into each other is because at Natural Trap Cave, the questions that I'm asking are, how did the Pleistocene extinction event affect megafauna? How did it affect the mammalian and other megafauna that were around, or the microfauna? So how did these things change? And in collaboration with my co-PI, Alan Cooper, we're looking at changes in genetics of these Pleistocene organisms, and I'm looking at changes in morphology in them. So we're doing this combined genetic morphological study. So I'll tell you guys a little bit about Natural Trap Cave, and I'll show you some cool field pictures, and hopefully that'll get you excited. So Natural Trap Cave is in Wyoming. It's basically in central Wyoming at the base of the Bighorn Mountains. It's just about 3 miles south of the Montana border. So you can actually see what this area looks like, or it doesn't look that different from what you guys have here, actually. We've got some nice foothills, and there's actually a lot of dinosaur fossils that come out of these sediments around here too. But this cave that I'm working at happens to be a Pleistocene sinkhole. So it's a trap of Pleistocene animals. Here it is. And what I really like to show people is you come up here and there's this little hill, and actually you're standing pretty close here with this sign, but if you're about 20 feet further back, you can't see the hole when you're standing there because of that little hill. And that's really important for how the animals got trapped, because a lot of the animals that are trapped in that cave are runners, they're open habitat dwellers. So basically they would be running along, they would run over this little hill, they wouldn't see this giant hole there, and in they would go. And the drop is 85 feet, and so it doesn't matter how, good you are at running. If you're falling 85 feet, you're going to die. You're going to go splat. We actually did a taphonomic study while we were out here, and also a physics study on how much body mass you have to have before you can survive that fall or not survive the fall in a natural trap. So this is an artist rendering by one of the original excavators of Natural Trap Cave, who is now a curator at the Los Angeles County Museum, Xiaoming Wang. He came out with me this summer, and he drew this rendition of bighorn sheep being chased by an American cheetah, and it looks like they missed the hole, or they missed seeing the hole, rather, and they fell into it. Looks like the bighorn sheep's trying to get out. He's not going to have a good time. And this cheetah's probably not going to have a good time either. So the critters that we have in this cave are the bighorn sheep right here, like I said. Those are probably the same species as the living bighorn sheep that are around there. We have the American cheetah, which is a close relative of the puma, but has the same sort of ecomorphology as the African cheetah. It's a runner. We have couple different species of horses. We actually have a cabalene-like horse, but we also have a stilt-legged horse. That's at Natural Trap Cave. We have quite a few bison. We have some camels. We have a couple of mammoths down there. And we also have this guy who's one of my favorites. This is the American lion. This is actually the skeleton from Rancho La Brea, but we have a couple of these guys down there. They're about a third again as large as a living African lion. So if one was standing next to me on the stage, his shoulder would probably be about the same height as my shoulder. And that's with him on all fours. So these guys were some honkin' lions. They weighed about 450 kilograms. That gives you an idea. All right. So here's a view of Natural Trap Cave from the other side. So you can see this hill here. This is my collaborator, Penny Higgins, staring down into the cave with the grate on. They put this grate on in the early 70s, in 1971, because it used to be a really great site for cavers to go spelunking in, and then somebody almost drove their car down there. So they had to put a grate on it to make sure that nobody killed themselves around that area. Yeah, you really can't see it at all if you're standing up here where this guy's standing. It's really hard to see it. You can see this big metal thing, but before this grate was there was nothing. You couldn't see a thing. So here's our team sitting around waiting for the cave to be opened. So this cave is actually owned by the U.S. Bureau of Land Management. We got a couple of BLM folks here. This guy is Brent Breithaupt. He's the regional paleontologist that helped us, and this lady here is Gretchen Hurley. She's the local person. She's a local BLM office person. And so this was our team. Our team was composed of several scientists and students, as well as several cavers who do this more or less all the time. And we really are glad that they were there because we really needed their help. And here's my co-PI, Alan Cooper, and here's me, in case I look quite different than I do today. So here's Alan actually doing the ceremonial opening of the cave. And this cave actually was excavated originally between 1971 and 1985 by a team from the University of Kansas and the University of Missouri, Larry Martin and Miles Gilbert. And they were down there for quite some time. When they originally had the cave open and they had all their excavations going, they had a scaffold that went from the top of the cave all the way down to the cave floor. Well, that scaffold has since been removed. And the only way we can get in and out now is by doing single rope techniques, rappelling in on a single rope and ascending out on a single rope with a technique called frogging. So our whole team did the single rope techniques. And we so nobody has really been down there. No scientific team had been down there since 1985. And so this really was a ceremonial reopening of the cave, and we were all super excited. And this guy standing right here, that's Xiaoming Wang. That's the guy who drew that, that cheetah chasing the sheep picture. And you'll see more of him. So here are Xiaoming and I getting ready to go down on the first day. And I really like this picture for a couple reasons. So Xiaoming was actually a graduate student on the original excavation. He did his PhD with Larry Martin at the University of Kansas. So he was part of the original team that went down into the cave. And it just so happens that Xiaoming was on my dissertation committee. So Xiaoming was part of my dissertation, and now I'm reopening this cave, and I had the pleasure of inviting Xiaoming out to come with me, and he had a great time because he was the only one on our team that had ever been down there before. Nobody else had ever been down there. So I thought this was 2 generations of scientists getting ready to make the re-inaugural opening of Natural Trap Cave. And we're wearing all of our caving gear, getting ready to go down. So here is me rappelling into the cave. This really cool thing back here is a defunct raptor's nest. Evidently, raptors used to nest in this cave quite frequently, and that's evidenced by all of the microfauna that we found in the cave, which appears to be raptor detritus. So all of the digesta that they didn't want, all the bones they didn't want, they threw it back on the floor, but it gives us an excellent record of all the microfauna that were around the cave at the time. But since they put that grate on there in 1971, the slats are too small for big raptors to get through. So these nests have all been abandoned since then. But they're still there, and they're pretty impressive. They're pretty big. I mean, we're at the same level. You can see how big this raptor nest is. So here's what it looks like to rappel down in from the bottom. The cave actually has quite a lot of light in it, much more than we expected. This is my friend Justin coming down on that rope. And you can see the light from the opening up here. This little dude is the counterweight for the door. He looks like a little caver going, woo. It's pretty funny. And this is a view from the cave, from the back of the cave, so looking up. So it's a beautiful cave. This drop is 85 feet, like I said. And you can see a guy up there on the ledge. And this down here is the excavation pit. And here's Alan holding one of our buckets. So this is how we got specimens and supplies up and down, was on this double pulley rope here with a bucket on the end, or we would hook other equipment, like we ended up getting these lights down here. We didn't need them, which was great. So that was our system of getting things in and out. And so here's a picture of the actual excavation pit. This is the pit that they dug in between 1971 and 1985 on that original excavation. And you can see here this guy is about 6 feet 3 inches. So you can see how tall the walls are. So they actually did about an 8 or 9 foot excavation pit. So pretty deep. They got down pretty far. And this is the zone directly below the hole, so the drop zone where all the animals would have fallen in. And actually you can see this ladder right here. It looks like a ladder to nowhere. This is actually still there from a little tiny rectangular pit that they dug down even further. That pit went down about 8 feet, and that ladder is basically stuck in there. It's about a 9 or 10 foot ladder, stuck in there with all the infill from the previous excavation in there. So it's unlikely that we will ever get back down there, but that's how far down the sediments go. They were pulling fossils out of that 10-foot pit. And so we only have 2 more years on my grant of digging here, and so I have a feeling that we're never going to get all the fossils out. Xiaoming told me that it would take someone 100 years to remove all of the fossils from this cave. So hopefully we're going to find a lot of good stuff over the next 2 years, and I'll show you what we found this year. So here's me and a couple of my team members, Justin Sipla, and a student of Penny. Her name is Jenna. We were excavating this pile right here. There's a bunch of horse stuff falling out right here, so we were trying to mitigate that deposit. And here's what that looked like. So for those of you who can't quite pick out what these things are, this right here is a horse tooth. So you can see it's a molar. It's got these, it's a high crown molar. You can see the little tiny rootlets down here. This is the proximal phalanx of a horse. So it's the first toe bone. Here we have some other toe bones and other hand bones of a horse. This looks like a vertebrae right here, a horse vertebrae. Here's another horse vertebrae, and here are a bunch more horse teeth. So this was sort of falling out of the side of the wall when we first got down there. So that's actually how we knew where to start digging. And this is the metapodial of a Pleistocene camel. So I don't know if any of you guys know, but camels and horses actually evolved in North America. This is their home continent. And then they went over to Eurasia and South America independently. But this is a Pleistocene camel. This is a cannon bone of a camel, so a metapodial. And that's my collaborator, Alan, holding it. And I'm going to show you guys a video of Alan doing some ancient DNA analysis or doing some ancient DNA sampling. There we go.
Okay, so this is a beautiful specimen in Natural Trap Cave. This is a camel, North American camel. That's where they evolved. As you can tell, beautiful specimen with classic camel toe, which makes us all laugh. Very, very big, much longer than any horse or bison. And what you can see inside here is how white and yellow, how unchanged the original bone is in the middle of that sample versus the dark staining around the outside here, which is coming from the soil. So that even though the outside of the material is being impacted by being in the cave, there's still this inner section of bone, that white yellow you can see there, that is in really good nick and should give us great DNA. So an unusual specimen, the camels, and fantastic to get a whole big leg bone like that out. All right.
So when you drill these things, they smell like modern bones burning. So they smell like burning bone, or if you've never smelled a burning bone, burning hair, that's what they smell like, suggesting that there's still a lot of good collagen in them, which is what we get the ancient DNA out of. That kind of material. If there's collagen, there's going to be DNA. That's basically the rule.
I'll save you like that. You still filming? Mm-hmm. If you're interested, this specimen here, right here, we found with that end sticking out, if you turn around there, just to the left of the bucket, about 1 foot to the left of the bucket, about 1/3 of the way up, sticking straight into the bank, we found the end there, and then pulled this whole camel right out.
So, so the ancient DNA is really, I'll wait for that slide. The ancient DNA quality that's coming out of these, so Alan did some preliminary ancient DNA on the stuff that was pulled out in the 70s and 80s, and he actually got pretty good mitochondrial fragments out of those specimens. So now what we're hoping is that because everything's been down in the cave, this cave is actually like a refrigerator. It's 40 degrees, even in the summer. It doesn't really get any warmer than that, and in the winter it's even colder, and it's very, very moist. So you can feel the air when you're down there. Because of these conditions, we're hoping that the bones that we're pulling out now, we might actually be able to get entire mitochondrial sequences and possibly even some nuclear fragments, which will give us an excellent picture of what these Pleistocene species genetic codes look like. So we'll be able to answer even more questions than we did with the stuff that was sitting in a museum for a long time. And because as it sits in the museum, the DNA degrades very quickly. So what we're doing now with the specimens that we pull out for ancient DNA is after we sample them, we ship them back on ice and we put them in a deep freeze until we are ready to sample them again. And so basically what we do is we sort of freeze them in time, more or less, so that we can actually keep them for posterity and resample those ancient DNA samples so that we have specimens that are not degrading in museums. And so I told you we did a little experiment, a little taphonomic experiment, plus a little physics experiment. We came down into the cave one day and found this poor little guy at the bottom in the drop zone. We named him Packy Le Pew. He's a pack rat, a North American pack rat, a neotoma. And so what we decided to do is that this guy, oh gosh, he probably weighs, you know, I don't know, maybe 20 grams, something like that. He's not very heavy. And so he didn't manage to make it when he fell 85 feet. So that doesn't bode well for something like us, or a horse, or a mammoth, that I imagine would probably splatter on impact at 85 feet. So this little guy didn't make it. So we decided to put him in a cage and do a little taphonomic experiment to see how fast he decays down in this sort of refrigerator environment. So hopefully when we go back out there in July, we will still see him. Something big hasn't snuck in from another entrance and dragged him away, hopefully. Whereas this little guy, I found him on the last day that we were down there. This is a Paramiscus, a little deer mouse. He fell in one night while we were up at the surface, and he survived. So the difference between a few grams between a pack rat and a deer mouse is the difference between life and death at an 85-foot fall. So this little guy actually survived. He was very cold and very scared, and we picked him up and put him in a bucket and sent him back up to the surface, and we gave him a little piece of apricot and a little thimble full of water, and as soon as he warmed up, he ran off into the bushes. As far as I know, he lived. Maybe he was just in shock and he died, but he was totally fine. So I thought that was a really cool physics experiment that was done while we were there. So this is my student, Adriana. She is washing all the bones that we got out of the cave, and so here are all the fossils that we have. Here's what they look like. So you can see all those horse toe bones here, a lot of horse teeth we did find. This is a horse jaw. You can see the incisors right here, and here's the symphysis. This is a piece of a horse metapodial. This is a radio-ulna of a large ungulate, probably a horse. But we also found some other really cool stuff. Don't have a picture of any of the cheetah that we found. We found a lot of the American cheetah. We found a lot of great postcranial elements. We didn't find any teeth of it, but we did find a piece of face of an American lion. I found it and I said, "Cat face!" And everybody was like, "What?" I was so excited. It's a piece of a maxilla with the incisors and some of the premolars, and then we also found a big chunk with the big P4 upper carnassial tooth of an American lion. So we found a few other bits of an American lion, a big humerus, couple of phalanges and some metapodials and such. But we found a lot of American cheetah, a lot of wolf, and a lot of horse. And so we do have some data on Natural Trap Cave that was taken previously by my collaborator, Penny Higgins. She did some stable isotope analyses of some of the critters at Natural Trap Cave, mostly the herbivores. And we're in the process of actually doing more of this right now. I sent her about 20 horse teeth that she's going to serially sample. So we're going to hopefully have some really good climate data from Natural Trap Cave very soon. But what you can see here is so this is the axis of oxygen, stable oxygen isotope. Down here we see colder environments. Up here we see warmer environments. And then this is the axis of stable carbon isotopes. Over here we see grazers, so this is the grazing axis. Over here is the browsers. So they're eating leaves and things as opposed to grass. And what we see here is, so bison are these red squares. Bison are right here. They are more grazing than browsing, and all the other critters that we have are actually more browsing, which is kind of surprising to me that Equus, the horse, would actually be more browsing than the bison. But that's what we find here, and we're going to be doing a lot more of this and looking at all sorts of different critters. So not just the herbivores, but we'd like to get a great idea of what everything was eating at Natural Trap Cave. And so now that I've talked to you about the site, I'm going to take about 5 minutes, maybe a little less, to talk to you guys about some of the wolves from Natural Trap Cave and what I'm doing with those. I'm comparing them with wolves from the Yukon, the Pleistocene Yukon, and also from modern localities to see how they're different and how they're the same. And so, with that, some wolves. So a lot of people ask me, are they dire wolves or are they gray wolves? Well, that was a really good question. Most Pleistocene sites, you don't see any gray wolves. As I explained before, they didn't really come over in mass quantities until after the extinction event, but Natural Trap Cave is actually an exception to that. It's one of the only Pleistocene localities that has large numbers of gray wolves, and as far as I can tell, not a single dire wolf. I have measured every single complete specimen from this cave, everything they collected at Kansas, in the 70s and 80s, everything we've done today or done last summer, and I have not found one evidence of dire wolf. So this is a gray wolf skull from the Yukon. So this is the kind of stuff they have there. Actually, as we speak, I am getting a shipment from the Yukon Museum of these gray wolf specimens. So I'm going to do some three-dimensional analyses on those. And this is a foot of a wolf that we got from Natural Trap Cave this summer. This is all of the hind metapodials, all the metatarsals, and we even got one phalanx. So this is kind of unusual. We hardly ever get anything in situ at Natural Trap. They found an American lion in situ in the 70s, but this foot was pretty much all together. So we think this foot belonged to one wolf. And so the way that I'm taking this data is I'm doing three-dimensional geometric morphometrics. So I take a microscribe, which is basically, it looks like a pencil on the end of this big mechanical arm, and I take data points, and it gives me a three-dimensional point for each data point I take, and I can compare those between individuals or between subspecies or populations, whatever you might want to call them, and compare them and figure out how similar or different they are. And this is just the rendering, the three-dimensional rendering in 2 dimensions of half of a wolf skull. So I digitize these crania of the larger Canis specimens, so anything that I know that is not a coyote, anything bigger than a coyote, I digitize those from the Kansas Museum. And then I also digitize, we also have data that a collaborator of mine, Anjali Goswami, took from the Field Museum of Modern Wolves. And I am in the process of getting these wolf skulls from the Yukon and measuring them. And so I'm going to compare these groups in a freeware package called Morphology with those three-dimensional points to see how different they are. And some preliminary analyses that I've done on this between the wolves from Natural Trap and the wolves, the modern wolves, suggest that the Natural Trap cave wolves are actually not morphologically significantly different from living wolves. So the wolf populations have not changed that much in 13,000 years, which is completely different than the coyotes. So I wasn't really expecting this result, but it's interesting that the wolf populations aren't changing. Like I said, they're so similar to modern wolves that there's no way they could be Canis dirus. They're all Canis lupus, so they're all the same species as the wolves we have around today. And I have a very small sample size right now, but hopefully that will change over the next 2 summers. And I had very few measurements initially, but I've gotten a lot more measurements in the last few months. So I'm going to rerun these analyses and see what I get with all the wolves included. So the next steps are to digitize those Yukon wolf skulls, digitize more modern wolf skulls to get a little bit more variation, see if we have a little bit more variation in the modern wolves that we can compare with, analyze everything, and hopefully plan to publish it this fall. And so that is the plan with the wolves, and I'm super excited about it because I really like wolves. They're an important species, and they're really rare in the Pleistocene, but they're really common at Natural Trap, and I'd really like to know what the conditions are that made that possible. So that's one of the things I'm going to be looking at with this project. And with that, I just wanted to thank all of my collaborators who've worked on these projects and papers with me. So these are sort of my co-PIs at the Natural Trap Cave site. All the rest of them are collaborators that have participated on papers with me that were talked about in this research. Here's my student, Adriana. And then I just want to thank my whole field crew from Natural Trap Cave who came out with me this summer and made it a success. And hopefully most of them will be coming out with me again next summer to find even more wonderful stuff. And with that, I just want to thank all of you guys, especially thank Francois for inviting me out. I've never been to Alberta, so this is my first time. It's very much like the Midwest of the United States, but it's beautiful. I love the Badlands area here, and I'm really glad to be here today. So thank you.